Hi, everybody. My name is Huzaifah Sheikh. Today, my presentation is about enhancing drug development through advanced NMR techniques, insight into molecular binding kinetics. This is from the Marlowe Lab at the University of Arizona College of Medicine, Phoenix. So, some background into our research. We are trying to affect protein-protein interactions, which are emerging as a promising strategy for drug discovery. The previous research indicates that the interaction between the focal adhesion targeting domain of focal adhesion kinase, a protein, with paxillin, a peptide, has been identified as a key interaction in melanoma. Our research focuses on using small molecules with the aim of bioavailability. So my personal background in this is I was very intrigued by the project because previously I was working over the summer in a cancer center, and it was an infusion center. So I was able to see how patients actually had to deal with IVs and how it could be uncomfortable or how it affected their body temperature. And it really made me more passionate about the subject. So the goal of the research was to improve iterative drug design and using HSQC as a biochemical assay to analyze small molecules. So in order to start the project, we worked on creating focal adhesion kinase, specifically the focal adhesion targeting domain. And we worked on protein preparation in N15 minimal media, and we used E. coli, and we did bacterial culture and growth. And this is our optical density curve to show that we reached saturation and induced protein expression to create our focal adhesion kinase. So we used our concentration of protein to determine our drug spikes. So we used a pure 660 assay to confirm concentration using bovine serum albumin, which is our standard. And then we created 1x protein stock at 10.5 milligrams per milliliter. And then we ran a Kumasi to confirm that we had the protein that we intended to have. We used several different steps in the process, including a nickel NTA pull down. We concentrated the product, pooled it using FPLC. So the methods of the research, why are we using 1H15N heteronuclear single quantum coherent HSQC in nuclear magnetic resonance? So HSQC reveals single bond coupling between protons and tagged nitrogen, and all amino acids, as shown here, have one NH bond, one backbone NH bond. And so what that does is it tells us much more detailed information about how the chemical environment shifts as opposed to whether the ligand is binding or not. So some previous research indicated that our small molecules were shown to be binding, and one of them using surface plasmon resonance had a tenfold increase, but the results weren't replicable. So we wanted to run HSQC to tell us much more about specifically where it's binding, how that chemical environment is shifting throughout the binding process. And it can tell us specifically which residues are moving when we bind our small molecule to our protein. So that's much more specific. It tells us a lot more information. As you can see, the nuclear magnetic resonance machines are there, and you can see specifically in our spectrum how our KD can shift based on specific residues. So it's a much more detailed and informational assay as compared to some other methods which we had tried in the past. So here we can see our spectrum overlays from our NMR, and we're able to analyze specifically our residues of interest. And so drug A is a hypothesized better binder, and we have our labeled residues G958, which are our residues of interest, and 959, L959. And as you can see on the spectrum, there's clear changes over the titration of the drug. So you can see these colorful changes. Drug A was a hypothesized better binder based on previous information. And as you can compare visually, which we do in chemical shift perturbation analysis as well, how these residues change over the course of the titration, with the lowest titration being in blue and the highest titration being in red. So we took our data specifically from the HSQC and NMR, and we ran chemical shift perturbation analysis of the peaks and line shape analysis as a measure of binding based on chemical shifts. So HSQC is a very data-rich experiment, and our chemical shift perturbation was not as conclusive as we had hoped, so we tried line shape analysis. And line shape analysis is a more detailed way of analyzing the quantum mechanics of the binding interaction. So these two forms of analyses can tell us different forms of data about our binding coefficient. So this is more detail about chemical shift perturbation analysis. As you can see, we have our 10-point titration, and we analyze it peak by peak, and it tracks the drifting chemical shift along one coordinate direction throughout the titration. It approximates fraction bound. So as you can see here, we can calculate our KD from this chemical shift perturbation, our approximate IC50 in a way. So we can determine that data from our chemical shift perturbation analysis. However, one of the drawbacks is that it might not recognize higher order kinetics, higher order binding kinetics, which is a problem we came across because we hypothesized that drug A was almost 10 times better than drug B. And so we expected there to be more movement, but we didn't see that. So we hypothesized that perhaps it was a higher order binding kinetic. And so we proceeded forward with a different form of analysis. So as you can see here, these are our interesting residues, G958, L959. We did the chemical shift perturbation of, and we analyzed our two-state KD comparison. So as you can see here, there's actually no significant difference. So this is where we came across that brick wall where we said, well, we were hypothesizing that one was better than the other, but we're not finding that here. And so as you can see, we have our KD values approximately similar, around 174 and 180, and then 190, and then an overlap in terms of standard error. And L959, G958, visually you can confirm that they're fairly similar, and these are our residues that we expected to move the most or change the most. So we proceeded with a new form of analyzing the data through Titan software. Titan created by another researcher analyzing NMR. Titan software, we were able to track the peaks and then run two-state analysis on drug A in G958 and L959. As you can see here, these are our overlays, and then we're tracking where the peaks are moving over the course of our titration. And then drug B here, once again, tracking the same peaks, trying to analyze what the binding kinetics are, what changes, or how our small molecule, the changes in the small molecule might affect specific residues or binding kinetics. So, and then from KD in chemical shift perturbation and Titan NMR, we also found that there was not a significant difference between binding kinetics. As you can see here, the KD is shown. So from that, we realized that we might need to run more, find more information about higher order binding kinetics. We weren't able to conclude, we failed to reject the null hypothesis in this case. Which data analysis works better? CSP analysis and line shape analysis both confirm the same conclusion, as well as previous data, which was showing that there was not replicable change in binding kinetics. So it tells us a lot of information about how to run HSQC and how we can change our experiments in the future to better show binding kinetics, especially higher order binding kinetics, which are in study or need further analysis. So some of the conclusions from this are our implementation of MATLAB in our workflow with HSQC, and we intend to create a crystal heat map of on and off rates of individual residues. So showing in our binding area which residues are changing can be very significant to the research and how we change our drug design in the future. So even if this didn't conclude with how the binding kinetics changes for these small molecules, we can also see how other changes of influence the residues of interest. So we're going to have further characterization of binding in the future and further iterative drug design. So during this research, I'd like to acknowledge my research mentor, Liam McCreary, in the Marlowe Lab at the University of Arizona College of Medicine, Phoenix. This is our work cited, so papers on Titan NMR and how we concluded which residues are which and how previous background showed information. Thank you.